Welcome back to AllAboutRx. Today, we're decoding gluconeogenesis. When your dietary carbs and liver glycogen stores run low, your body doesn't just stop. It starts making its own glucose. This is absolutely critical for glucose-dependent tissues, so let's dive right in.
So picture this, you're 10 to 18 hours into a fast and boom, your liver glycogen is completely wiped out. Your body is thrust into a literal metabolic crisis. It has to scramble to feed obligatory tissues. I mean, your brain, your red blood cells, the kidney medulla, they can't just switch off, right? They demand glucose, and your body has to figure out how to make it on the fly. Here's our quick roadmap for today's explainer. 1. The fasting glucose crisis. 2. Gathering non-carb precursors. 3. The 3 glycolytic roadblocks. 4. The 4 bypass reactions. And 5. A high-yield quick recall to lock it all in for exam day.
All right, Section 1. The fasting glucose crisis. The primary problem we're facing is that the body is completely out of dietary glucose and glycogen stores. Now, gluconeogenesis is the ultimate physiological survival mechanism. It steps in precisely when those carbs and stored glycogen are insufficient. Since there aren't any carbohydrates left, the body has to get incredibly resourceful and synthesize essential glucose entirely from non-carbohydrate parts. It's basically building a house without any bricks. So where does this magic actually happen? Primarily in the liver, and to a slightly lesser extent, the kidney. If the liver didn't step up to maintain blood glucose levels, fasting would be an immediate disaster for all those dependent tissues we just talked about. Moving right along to Section 2, gathering non-carb precursors. To build this new glucose, the body has to scavenge raw materials from various tissues. You really need to know where these non-carb parts are coming from. Think of them in 3 main categories. First up, glycerol. We get this from the hydrolysis of triacylglycerols in your adipose tissue, your fat stores. Second, lactate. This is efficiently recycled from exercising muscles via the Cori cycle. And third, amino acids. During fasting, tissue proteins break down into glucogenic amino acids, which become your absolute major source of raw material. Okay, Section 3, the 3 glycolytic roadblocks. To make this glucose, the liver essentially runs glycolysis in reverse. But trust me, there are some massive roadblocks in the way. Out of the reactions from pyruvate back up to glucose, 7 of those glycolytic enzymes are a reversible two-way street. The body can just push the reactions backward, no problem. But 3 steps are totally irreversible. You can't just drive backward down a one-way street, right? And here is the high-yield secret you absolutely must remember. To get around those irreversible steps, hexokinase, PEPCK1, and pyruvate kinase, the body has to use unique, specialized gluconeogenic enzymes as gatekeepers. If you want to synthesize glucose, you have to take these specific metabolic detours. Which brings us to Section 4, the 4 bypass reactions. Let's break down these highly specialized bypasses that solve our metabolic roadblock problem. When you look at the big picture pathway from pyruvate all the way up to glucose, the reversible reactions are a breeze. But we have to take distinct, pretty energy-intensive bypasses to maneuver through the rest of the maze. Let's walk through those turns. The very first critical bypass is the carboxylation of pyruvate to oxaloacetate, or OAA. This happens deep inside the mitochondria. The enzyme pyruvate carboxylase does the heavy lifting here. But, and this is huge for your exams, it requires 2 things: biotin as a coenzyme and acetyl-CoA as an activator. Acetyl-CoA basically acts like a metabolic alarm bell, telling the cell, hey, we urgently need more OAA. But then we hit a snag. OAA is trapped. It physically cannot cross the inner mitochondrial membrane to get out into the cytosol, where the rest of this whole process happens. Enter the malate shuttle. OAA is temporarily reduced to malate. Malate easily sneaks across the membrane, effectively ping-ponging into the cytosol. Once it's across, it reverts right back to OAA, which importantly generates NADH in the cytosol, something we'll definitely need for the reverse glycolysis steps later. Once we're successfully in the cytosol, our second unique enzyme, PEPCK, takes over. PEPCK uses GTP hydrolysis to convert OAA into phosphoenol pyruvate, or PEP. So by using pyruvate carboxylase and PEPCK as a tag team, the body has officially bypassed that very first glycolytic roadblock, pyruvate kinase. Moving up the chain, we hit the next major detour, fructose 1,6-bisphosphatase. This bypasses PFK1. Honestly, this step is the primary regulatory site of the entire pathway. Think of it like a seesaw governed by the cell's energy state. It's stimulated by high ATP, meaning the cell is energized enough to do the hard work of building glucose. But it's strongly inhibited by high AMP, which screams energy poor state, meaning the cell needs to conserve its resources, not spend them. Finally, we reach the top, the last bypass. Glucose 6-phosphatase removes the final phosphate, bypassing hexokinase to release free, usable glucose straight into the blood. And here is a vital, high-yield fact. This specific enzyme is a feature entirely unique to the liver and the kidney.
Let's wrap things up with Section 5, High-Yield Quick Recall. We're going to rapid-fire the absolute must-know facts for your exams.
Lock these details in right now. The main sites are the liver and kidney. The pathway relies on biotin and acetyl-CoA as essential activators for pyruvate carboxylase. GTP is what drives PEPCK. F16B PICE is the major regulation point, inhibited by AMP. And for your substrates, never forget that the Cori cycle is your primary lactate recycler. And to tie it all together, always remember the highly specialized nature of the liver and kidney. Because they are the only organs with glucose 6-phosphatase, they are the absolute sole providers capable of releasing free glucose back into circulation. They literally keep you alive during a fast. I'll leave you with this final thought experiment. If your exercising muscles also had glucose 6-phosphatase, how would that alter the entire design of our blood glucose survival mechanism? If muscles could just release their stored energy into the blood instead of keeping it for their own contraction, would we even have the immediate localized energy needed to, say, run from a predator? It's pretty amazing how perfectly this system is balanced. Anyway, thanks for studying with me on AllAboutRx. Don't forget to hit subscribe and check the link in the bio for these high-yield notes. Keep studying hard, and I'll see you in the next lecture.